Okay, so I was team
Are you
10.
on mute?
Mm, I think most people can hear me. I am unmuted.
Hear you.
Amy, can you hear me?
Yes, go ahead.
Okay, so I am team 10. Our goal was to prepare for eventual sequencing of clinical COVID samples, well, SARS-CoV2 samples here at UAB, and that we foresaw the need to then analyze those, both take the genome and build a patient-specific genome, and then place that into a global context. What geography does this strain most resemble? Where is it most likely to have come from? So we can start doing some epidemiology around transmission of virus and virus clusters, especially if we wanted to do some work in the hospital about transmission inside the hospital or in any particular setting. This type of tool of sort of genomic epidemiology is extremely useful and has been used in many other countries and other cities to help control the virus. So we thought we should have these tools up and running here before we actually had the sequences. Clearly, this is an effort we should have started earlier, but now is a good time. So we called ourselves novel to global. It's the novel sequence that we observe here into the global context. Let's see. This outlines sort of our goals. And we wanted these pipelines to be as automated as possible. This is pretty standard technology, so we wanted to make it fast and reliable, particularly in terms of updating our reference panel from the global databases, since that changes with startling regularity. But even the parts of the pipeline that involve building the patient-specific sequence and handling the sequencing are relatively easy to automate. So before the hackathon, we'd already been involved in this process in that we'd built a pipeline to process SARS-CoV2 samples that had been grown in the lab in cells to demonstrate as part of a QC effort. And we'd successfully aligned that against the reference genome. So we had that technology and pipeline as an automated pipeline already up and running. So this is sort of the workflow we were hoping to accomplish. And I don't know if you can see my cursor. On the left is what we have to have locally, pulling stuff from the client, aligning it, creating a consensus genome. On the right-hand side is pulling from the global data banks, and then building a global reference, alignment, and phylogeny, and then merging that with our local data to produce a report of where our local data fits into that global context, and then the visualizations, interactive visualizations of that. We're basing a lot of this idea on this nextstrain.org, which is a set of public tools that have been made available to do this, and they've set up interactive websites not only for the novel coronavirus, but for several other pathogens, and they've provided open source tools to help other people do this. So this is sort of our plan A, is to try and use their tools to implement this with our data. These were our priorities. We only had one person on the team, so that kind of limited the scope of what we could take on, but we did have an expert, Elliot Lefkowitz, who has a lot of history with viral phylogeny. So our first priorities for the hackathon were getting the global data in-house in the right formats, getting the processing steps online to create the global reference. On Monday, we got the reference data downloaded. I'll note that there were about 5,400 samples on Monday. Today, if you look in that same database, there's 7,600. So this is a moving target, which is why we want it automated. We had a plan A, which was to use that Nextstrain public pipeline, which is available as a Docker container and in several other formats. And plan B was to do a lot of this locally with our own tools. We made the decision to start with the local plan B approach because we were a little unsure whether we could get Nextstrain or implemented in the time frame of the hackathon. So we spent Monday getting the reference data downloaded, reformatted, and then we started running Muscle, which is the general aligner. And that ended up taking enormous amounts of resources, and we ended up essentially running out of time because it's too slow to use on a data set this large. We could do it on 100 genomes, but we couldn't do it on thousands of genomes. So that was Monday, and a lot of that stuff ran out of time and died over the evening between Monday and Tuesday. So on Tuesday, we continued on our plan B. After more research, we found a different aligner that is more high throughput and that had a new version specifically for doing these COVID alignments very fast. And in fact, that only took minutes to run instead of hours. So that was a big step forward. We haven't validated that against the muscle alignments that did eventually finish later on Wednesday. But we'll move forward with that. And it actually turns out, once we had that in hand and we knew we had a plan B in place, we went back to trying to do Nextstrain and using that public pipeline. It turns out that actually uses that MAFT aligner with this fast mode. So we spent most of Tuesday on getting the Nextstrain code in-house. It's provided as a Docker container, which we were able to get up and running, at least passing its self-tests inside of SnakeMake on Shihai using the Singularity package to run the Docker container. The next step was preparing the metadata. So the information about all those public genomes, where did they come from? What's their geography? Geography is always a very difficult thing to code, lots of inconsistencies. So we pulled down the code from the metadata from NCBI that we pulled down the first day. And we ended up having to write scripts to reformat that and process it so it would match what Nextstrain expects. Nextstrain is sort of more set up to pull stuff out of a different database that uses a slightly different format. So we got that all prepared, and then we were laying out the files and the data in the way that Nextstrain requires and trying to get the Nextstrain pipeline actually up and running. So we got partway through that. We got the tutorial. They have a Zika tutorial that is supposed to be your training set that we got mostly to work. And we think we have all our files laid out correctly, but we're still having trouble getting the build to run from end to end to actually produce the interactive global alignment. So that was a little disappointing. We made a lot of progress. We got a lot of tools installed. In doing various steps of the process, we had to evaluate multiple tools and install them and quantify them. So we were able to eliminate tools, but it took a while to work through all that. So in the end, we have a good groundwork set up, but we didn't actually finish building a interactive visualization that we could actually hand to somebody yet. So the tool is not finished. So in our conclusion, we moved really far forward in 2 days. This was a great opportunity to really focus on something for a while and move things forward. By being a little bit conservative and trying to do the sort of known tools, things we were comfortable with the first day, we wasted quite a bit of time that we could have used probably if we'd just gone for the Nextstrain from the get-go and focused on that tool set exclusively. But we were trying to sort of balance our efforts in an effort to make sure we got something to work. And it's interesting on how fast things move. I've been getting up early in the mornings to participate in the Oxford Nanopore London Calling conference in London's time zone the last 2 days. And I saw a great talk by some folks in Connecticut who had essentially done just this. And that resulted in a Cell paper a few months ago, well, a month or two ago. So they were talking about that. And essentially they just sequenced all of their local clinical samples and used Nextstrain and some other tools to create visualizations. They did some additional work, epidemiological work that was quite important in terms of showing that transmission was coming in from other states in the US more than it was from internationally, and doing other important work in terms of explaining how the outbreak in Connecticut was unfolding. But it's quite impressive that something that we're sort of taking on as a hackathon was only a month or two ago, something that was made a Cell paper. And I think looking back, if we'd been reading the news in January, which we were, we might have wanted to put more effort into getting all these tools ready so we'd be ready to jump on this when it arrived, since we could all see this coming. And we wish, for those of us who analyze sequences, it would be much more exciting if UAB had started sequencing people much earlier on in all this. Because then we'd have more things to play with and we could have interesting results. But that's where we are. So it was very good to have the time to focus on this and access to experts like Elliot to advise. But in only 2 days and only one person in our team, we weren't able to make as much progress as we'd hoped to make. So that was a little disappointing. And I think I'll take questions then.
Thanks. We have one question. Why didn't Muscle work?
Muscle worked. It's just slow. It's a single-threaded application. And so the ability to parallelize it is limited. I think there is an MPI version, but trying to get MPI version of Muscle up and running was going to be a bit too, that was sort of our last fallback plan if we couldn't find anything else. So it works. It just takes inordinately long. It ran for 12 hours before it ran out of time and got killed by the scheduler on the full data set. Well, not even the full data set. That's 5,400 genomes. We removed the duplicate genomes, and that left 4,700. and that was just too many for muscle to do in that time frame. It's a testament to the power of parallelization and optimization that a version of MAFT, which is a kind of similar program that uses multiple cores and had some optimizations for doing closely related viral genomes, could do the same task in minutes. Muscle wasn't making any assumptions, or wasn't able to make assumptions about the fact that these genomes are so closely related, so it was using a much more general purpose algorithm.
There's just a comment that the publication of similar results in Cell shows significance of having the infrastructure in place before the pandemic begins. That's always a good thing. Another question, how many sequences were used for MSA?
We started, the MSA that we're going to go forward with has all, well, the one that we used in the hackathon has the 5400, including the duplicate, the 1,000 or some that actually turn out not to have any variation in them, that they're just additional data points with additional geographic information or patient information. If I was to rerun the pipeline today, it would be using 7,000 sequences, because they've added over 1,000 sequences to the data bank in the last week to NCBI. Which is one of the reasons why we wanted to focus on making this a highly automated pipeline, so we could easily keep this up to date, because doing it manually is not going to be tenable without having a lot of people to work on it.
What would you do differently next time?
Like I said, it would be good to have a few more people on the team to allow us to explore more options at once, because we're sort of limited by me exploring things serially. But I think having more time to research tools before the actual coding started, so we would not waste as much time on tool choice. So, for example, if I'd had time to spend a lot more time reading up on Nextstrain and the details of that before the hackathon started, then I could have spent more time in actual implementation. So that is just available time to prepare and study before diving in, and just, so to speak, person hours of resources.